Welcome back to Anatomy and Physiology on Catalyst University. My name is Kevin Tokoph. Please make sure to like this video and subscribe to my channel for future videos and notifications. In this video, we're going to discuss three mechanisms of capillary exchange. So here's a capillary bed right here. We know that it has a meta-arterial that's going to supply it. There is a thoroughfare channel, but these things up here where we see all these arrows, these are our true capillary beds. And so if we're talking, of course, about exchange of nutrients with tissues, then we have to get blood flow into these capillaries. And then, of course, we're going to get oxygen and glucose movement from the blood to the tissues, and then conversely waste products should move from the tissues into the blood. And all this exchange is going to happen at these true capillary beds. We talked about that in the previous video. Now, what are the mechanisms by which capillaries facilitate nutrient and waste exchange? Well, the first two are pretty straightforward. The first one is just simple diffusion. Okay, this is just means by which the particles move through the membrane. Okay. So, for example, oxygen is, of course, going to diffuse from the lumen of the blood vessel, that is the capillary, into the tissue, right? And eventually into the cell, right? I mean, that makes sense. We know oxygen has to move from the blood into the tissue. Conversely, carbon dioxide, which is produced by the tissue, is going to have to move across here into the blood. Those things we covered in previous videos, so hopefully you've got this. Glucose can actually move through spaces between the capillary cells. For example, if we were to look back at this slide right here, even in continuous capillaries, there are clefts between the individual simple squamous cells that make up the blood vessel, that is the endothelial layer. And so certain molecules like glucose can actually squeeze between those. Okay? But we're going to kind of lump all that together as simple diffusion because it's movement of particles and gases from high concentration to low concentration. So that's pretty straightforward. The second one is as well, vesicular transport. So for example, we can get things like pinocytosis or endocytosis and even exocytosis of certain things. Right here we're probably showing endocytosis or pinocytosis, but the reverse can also be true. We can actually move things in vesicles from the cells or tissues into the blood. Okay? And that's vesicular transport, which of course is an active process. It requires ATP. But these things are pretty simple. The most complicated of these topics is what's called bulk exchange. And bulk exchange depends on pressure differences between the arterial end of a capillary and its venous end. And we need to look at these pressure differences because the pressure difference is going to actually tell us what direction fluid is going to move. So if we look at these capillaries, we can just kind of follow one of them. Let's say this one right here. Okay? If we look at one end, it's still a capillary, but it's what we call the arterial end of the capillary. Okay, it has a higher pressure on this side. As you follow it through to where it intersects back with the thoroughfare channel, we get to what's called the venous end of the capillary. And the venous end is still going to have a pressure, but it's going to be lower than it was at the arterial end. Now, one key here that you need to remember throughout your study of the cardiovascular system, and really every system, is that particles and gases and liquids, they always move from high to low. So if we're talking about liquids like blood, it's always going to move from high pressure to low pressure. So the arterial end of the capillary ought to be a higher pressure than it is at the venous end. Okay, and so the blood is going to move from the arterial end to the venous end. So yes, it's important to understand that blood flow is unidirectional from arterial end to venous end. But also think about this. It's not just that the fluid is moving through the vessel like this from arterial to venous end, but because of the pressure inside the capillary, sometimes fluid might actually move from the capillary into the interstitial area, or then in some cases it might move from the interstitial area back into the capillary because of pressure differences. And it's not pressure differences within the vessel. We're talking about pressure differences between the inside of the vessel and outside the vessel. And those differences are going to contribute to what's called bulk exchange. So in this slide right here, we're going to talk about that bulk exchange in more detail. So here is a sample capillary right here. We have the arterial end of the capillary and the venous end. So this is all capillary, but there's different regions of it. Okay? So let's talk about the major forces or pressures that are going to be acting on the vessel wall. Okay? And we're talking about pressures that are inside the capillary. The first one is just called the blood hydrostatic pressure. We're going to call this HP with a subscript B to denote it's the hydrostatic pressure due to the blood. And this is typically what we just call blood pressure. So when you measure somebody's blood pressure, you're actually measuring a blood hydrostatic pressure. Okay? Notice at the arterial end, the blood hydrostatic pressure is about 35 millimeters of mercury. On the venous end, the blood hydrostatic pressure drops to about 16 millimeters of mercury. Okay? Now, what blood hydrostatic pressure is, anywhere in the vasculature that is it's really just proportional to the force exerted by the blood as it presses against the vessel wall. Yes, blood is moving in this direction from left to right as I've drawn it, from arterial end to venous end, but as it's moving through, it's exerting a force against the vessel wall. Okay? And so it's pushing from the blood against the wall. So this arrow right here, this is denoting the direction of the pressure. That is the blood hydrostatic pressure. It's pushing from the blood against the wall. Okay? And then we have another force or pressure called blood colloid osmotic pressure. We abbreviate this COP with a subscript B. Sometimes this is called oncotic pressure. Now, this pressure moves in the opposite direction, or it tends to move in the opposite direction. So inside the capillary right here, we have proteins, such as plasma proteins like albumin and so on and so forth. And proteins like water. Okay? So proteins are actually going to create a reverse pressure. That is, proteins in the blood right here are going to create a reverse pressure. Okay? They're going to actually want to bring water into the vessel, into the capillary. And so their pressure direction is actually going to be this one right here. It's going to actually tend to want to move things from the interstitial area into the capillary. So at any point in this capillary, we have two competing forces. We have the blood hydrostatic pressure and the colloid osmotic pressure. The blood hydrostatic pressure, remember I mentioned that its direction is from the blood out into the interstitial area. So blood hydrostatic pressure is going to tend to want to force fluid out of the capillary and into the interstitial area. However, in the reverse direction, blood colloid osmotic pressure is going to tend to want to move fluid from the interstitial area into the blood. And that's because we have proteins in the blood, and the proteins like water. So they're kind of attracting the water, so to speak, and wanting to pull the water from the interstitial area into the blood. Okay? Now, notice at the arterial end, the colloid osmotic pressure is about 26 millimeters of mercury. Also notice that as we go to the venous end, the colloid osmotic pressure doesn't change. It's 26 millimeters of mercury wherever you are in the capillary. What changes is the blood hydrostatic pressure. At the arterial end, it's 35 millimeters of mercury. At the venous end, it drops to 16. So we have a pressure gradient. We have a pressure gradient that is between the hydrostatic pressure and the colloid osmotic pressure. Now let's look at this end, the arterial end. Which one is greater? Well, just looking at these numbers, it's clear that the hydrostatic pressure is greater. And which direction did the hydrostatic pressure want to force fluid? Well, it's exerting a force from the capillary to outside in the interstitial area. So this hydrostatic pressure is going to want to force this fluid from the blood into the interstitial area. So this direction. Whereas the colloid osmotic pressure, being pressure that forces water into the blood to satisfy the plasma proteins, it's going to pull fluid from the interstitial area into the blood. And if you look at the relative sizes of these arrows, that's done to represent these numbers. And so because the hydrostatic pressure is greater, there's going to be a net movement of fluid from the capillary into the interstitial area, but only at the arterial end. And so that process of moving fluid from the capillary lumen, that is from the blood, into the interstitial area, that is called filtration. Okay? So we're going to see net filtration on the arterial side of the capillary. Okay? And what that filtration is going to do is it's going to move fluid out into the interstitial area, but with that fluid it's also going to bring nutrients. It's going to bring oxygen. It's going to bring glucose. It's going to bring amino acids. It's going to bring all sorts of stuff. Okay? So by a mechanism distinct from just diffusion or vesicular transport. It's due to the pressure difference. And because the hydrostatic pressure is greater on this end, it's going to drive fluid, bulk fluid, into the interstitial area. And again, that fluid with it brings all sorts of nutrients and so on and so forth. Okay? But filtration, that process is going to occur at the arterial end. Now, as we move through the capillary, so as we're moving through this right here, we notice that it turns into what we call the venous end. And I mentioned that the venous end has a lower hydrostatic pressure. So as we go from the arterial end of the capillary to the venous end, we mentioned that the colloidal osmotic pressure remains unchanged. However, the hydrostatic pressure drops from about 35 millimeters of mercury down to about 16 millimeters of mercury. And so now let's look at the venous end and see if the direction of bulk flow actually changes. And in fact, it will. Again, our colloidal osmotic pressure of 26 millimeters of mercury, that's this larger arrow right here. And it's, of course, going to force fluid, or tend to force fluid, in a direction from the interstitial area into the capillary. Now, if we look at the hydrostatic pressure, relatively speaking, it's a smaller number, 16. So it's a smaller arrow. And it would tend to force fluid from the blood into the interstitial area. But again, if we look at the relative sizes of these numbers, we see that the colloid osmotic pressure is stronger on the venous end. And so, because this is stronger, we're going to actually have a net movement of fluid from the interstitial area back into the capillary. Okay? So, some of that fluid is filtered at the arterial end, and it brings with it nutrients and all sorts of things like that. But then when you get to the venous end, some of that fluid actually ends up being reabsorbed. Okay? And so when that fluid is reabsorbed, it'll bring with it things like waste products, like carbon dioxide and so forth. Okay? Yes, carbon dioxide can move from the interstitium into the blood through simple diffusion. Yes. But this provides another means to get a lot more of it back into the blood. So reabsorption occurs because the colloid osmotic pressure in this region is greater than the blood hydrostatic pressure. And so you get bulk movement of fluid back into the capillary. And of course, it's going to bring with it waste products like carbon dioxide. Okay? Now, hopefully this process makes sense. What you should do is make sure you understand that filtration is occurring on the arterial end, and that reabsorption of fluid is occurring on the venous end. Understand that both of those are forms of bulk flow, and understand why those occur based on pressure differences. Now, you may not have to memorize these exact pressures, and you probably shouldn't because they can change depending on what source you're looking at. But the actual relative sizes of these are what are important. On the arterial end, the blood hydrostatic pressure is larger, and so you get filtration. On the venous end, the colloid osmotic pressure is larger, so you get reabsorption. Okay? Now, what's important to understand about the fluid that's reabsorbed is it's not all of it. It's not all the fluid that's reabsorbed. Only about 85% of the fluid that's filtered is reabsorbed. So if you look at the amount of fluid that gets filtered on the arterial end, only about 85% of that fluid is reabsorbed. The other 15% of it remains in those tissues. Okay? And that remaining fluid can actually be returned back to the blood via the lymphatic system. So, 85% of it's reabsorbed. The other 15% of it that ends up in the interstitial areas, it doesn't go into the cells because that would cause them to burst. Rather, it's returned back to the blood via the lymphatic system, which we've covered in another playlist, or at least we will cover. Okay? Now, one other thing I want to mention before we conclude the video about this percent of fluid that's reabsorbed, it's very important for the pressure. Okay? Notice that the arterial end has a higher hydrostatic pressure than the venous end. Remember what hydrostatic pressure is. It's the force of the blood pushing against the vessel wall. Well, the question I want to ask you is, why is the hydrostatic pressure on the venous end lower? Why is it lower? One of the contributing reasons why it's lower is because, notice, only 85% of the fluid that's filtered is reabsorbed. So, remember, blood volume is proportional to blood pressure. If you're only reabsorbing 85% of the fluid, then you have a lower blood volume in this area and therefore a lower blood pressure. And that's actually one of the major reasons why the venous end of the capillary is a lower pressure than the arterial end. And also another reason why the venous system in general is lower pressure than the arterial system. Okay? So hopefully that makes sense. Please make sure to like this video and subscribe to my channel for future videos and notifications. Thank you very much.